Hi there. My apologies for not being in class today, but I had a personal issue that came up and had to be taken care of, and I tried to get it taken care of before class started, and I was unable to do that, largely out of my own control. Anyway, what I want to do in this screencast is basically take you through the introduction that I would have taken you through in class today. For the next 3 weeks or so, we're going to be talking about animal diversity, form, and function. And today was intended to just kind of get you introduced to the groundwork. So by the end of this 3 weeks of learning, you should know the common features and major evolutionary innovations that allowed animals to diversify and become what is the most diverse kingdom of organisms on the planet. Not counting bacteria, which we just don't know enough about yet to know what their true diversity is. You should know how the animal body develops from zygote to gastrulation and what the implications are that for currently the way we classify the major groups of the animals. And then we're also going to do a similar thing like we did with the plants, in that we're going to go through and familiarize ourselves with the major phyla of animals as well as relevant aspects of their biology. And just like with the plants, I'm going to try and organize this on the basis of the movement of animals out of marine environments and into terrestrial environments. So when we think about the shared features of animals, animals are heterotrophic. They feed on other organisms. They are not autotrophic. They do not produce their own food. They will eat other organisms that do produce their own food, like plants, but they can't do it themselves. They're multicellular, which means that they're made up of many cells as opposed to single cells. The single-celled heterotrophs are the protozoans. They have no cell walls, but animal cells are held together in terms of their shape by, in some cases, extracellular proteins such as collagen and elastin, and then they're also held together by intracellular proteins like tubulin and actin. And these proteins inside the cells basically act like an internal skeletal structure that holds the shape of the cells. Animals are active movers. Most animals are capable of moving from place to place, but there are quite a few animals that are sessile. But even if they're attached to a fixed position, that's what the word sessile means, even if they're attached to a fixed position, their bodies are capable of moving in ways that the bodies of plants, for example, are not capable of moving. They're very diverse in terms of their size. Some of them are so small that you can't even see them with the naked eye, all the way up to the size of the blue whales, which are the largest currently living organisms. Most of the animals are invertebrates, and even though we like to think of vertebrates a lot because we also are vertebrates, they only make up about 60,000 species of the 1.5 million total described species of animals that are currently known. Animals live in a diversity of habitats, all the way from the bottom of the ocean to the tops of mountains. Most of the phyla are marine. Fewer phyla are in freshwater, and fewer still are terrestrial. So when we think about the major groupings, the big groupings of organisms, which is what phyla are, most of those phyla are marine. But within the terrestrial phyla, which there aren't very many of, that's where most of the diversity of species is located, is in those terrestrial groups. And so don't get those two things confused. There are a lot of phyla that are marine that don't have very many species in each of the phyla, and then there are a relatively few number of phyla that are terrestrial, but those phyla tend to have a lot of species in them. And sexual reproduction is the norm, but there are plenty of exceptions to that normal rule. Animals go through an embryonic development that is unique to animals and is different from the development of plants. A zygote grows into a multicellular individual via mitosis, but it does it in a very different way than plants do. And we'll see what that looks like later on in this lecture. Most species have larval forms that look different than the adult, and these larvae also occupy different habitats compared to the adult. One of the reasons that this might have evolved was just to reduce competition within a species by basically having larvae do one thing and having the adults do a different thing. And then we see tissues developing in animals. And we've talked about tissues in plants, but tissues also occur in animals. And all of the animals except sponges have their cells organized into tissues. And once again, tissues are just cells that are of similar morphology that are organized into units that perform a particular task. And just to review this, you've seen this in lab. The major tissues that we deal with in animals are the epithelial tissues, the muscle tissues, connective tissues, and nervous tissues. And remember that each of those tissues is defined by a particular characteristic. In the case of epithelial tissues, those tissues are tissues that either line the outside of the body of an organism or line inside cavities, internal cavities of an organism. So epithelial tissues, their fundamental nature is to line cavities or line surfaces. And the other tissues have similar things, which once again go back and review your materials from the first week of lab from this semester. So, as tissues develop, all of our tissues develop from a single cell, the zygote. When you were first conceived, you had an egg from your mother and a sperm from your father, and those two things came together to create a diploid zygote. And that zygote, that single cell, turned into all of the different cells in your body. It turned into your skin cells, it turned into your bone cells, it turned into your brain cells, it turned into the cells that make the coloration in the iris of your eye. They turned into blood cells. And so this is a situation which is referred to as totipotency, or those cells are referred to as being totipotent. And what that means is they can just become any type of cell. But as an embryo develops, an animal embryo develops, the cells eventually undergo differentiation in that those cells that could become any type of cell, they differentiate into the specific type of cell that they will ultimately become. And so you have these stem cells that could become anything. There comes a point in development when those stem cells can now only become muscle cells, or other cells can now only become nerve cells. And once differentiated, they only produce new differentiated cells via mitosis. You can't have a muscle cell that then is capable of producing a nerve cell. Cells of sponges are different in that they can de-differentiate and redifferentiate as needed. But it's also true that sponges have the least amount of differentiation of all of the groups of animals. So that's one of the trade-offs of complexity, is that once you start down the developmental pathway, it's difficult to change that fate of those cells. We also see the evolution of symmetry in animals. We have asymmetric animals, of course, but we also have radially symmetric animals and bilaterally symmetric animals. These are the definitions of the two types of symmetry. And one of the advantages of bilateral symmetry is it allows an animal to move through the environment in a particular way. And then radial symmetry is believed to have been evolved from bilateral symmetry, but the easiest way to understand radial and bilateral symmetry is to actually see it in organisms. In radial symmetry, if you cut through the middle of this organism in any plane, it divides it into two halves that look alike, and it doesn't matter what the plane is. In bilateral symmetry, only when you divide the organism in the sagittal plane do the two halves of the organism look alike. If you divide it in the frontal plane, it does not look that way. And if you divide it from dorsal to ventral in the transverse plane, the two halves do not look like mirror images of one another. And so that is the feature of bilateral symmetry, and it gives a directionality to the body that, for example, the sea anemone does not have. And then another thing that happens with animals is we have the evolution of body cavities. Over time, during the early development of the embryo, you begin to have the various cells start to differentiate into what we call endoderm, what we call mesoderm, and what we would call ectoderm. You can think of endoderm as the cell and tissue types that occur at the very core of the body of the organism. This would be the digestive system in us. The mesoderm lies between the endoderm and the ectoderm being those tissues that form the outer portion of the body. And then the mesoderm, things like muscles and bones, that's what makes up the mesoderm. The mesoderm gives rise to muscles, connective tissue, and things like that. So the ectoderm is the outer skin and parts of the nervous system of organisms. Endoderm is generally the digestive system, and then mesoderm is everything in between those other two layers. As this develops, you can have the formation of internal body cavities, and these internal body cavities accommodate the evolution of complex body organs. So when we think about the way the body of a planarian, a flatworm, is arranged, we have an ectoderm on the outside of the animal, we have an endoderm on the inside of the animal, and all of the space in between the ectoderm and the endoderm is filled up with mesoderm, muscle basically. In pseudocoelomate, so this is an acoelomate organism in that there's no internal cavity except the digestive cavity itself, which is not really an internal cavity as much as it is a tube that goes through the body of the organism. In pseudocoelomate organisms, you have an ectoderm on the outside, then you have mesoderm that is attached to that ectoderm, but that mesoderm doesn't come into contact with the endoderm. So the space between the mesoderm and the endoderm that is inside the body of the organism is not lined on all sides by mesoderm. It's only lined on one side by mesoderm. This is the pseudocoelomate condition. This is what we see in roundworms. And then by the time we get up to annelid worms, we see that there's an ectoderm, there is a mesoderm that has grown around and is reflected upon the endoderm, so that this cavity that is inside the body of the animal is actually lined on both sides by the mesoderm. This is the difference between being acoelomate, being pseudocoelomate, and being coelomate. Coelom basically just means opening or space. We used to think that these three characteristics, these three types of body cavity, had phylogenetic importance, but now that we have a better idea of phylogeny, we have discovered that, for example, the pseudocoelomate condition has evolved multiple times over the course of evolutionary history, and we'll see this when we get to the phylogeny at the end of today's material. So what happens with the development of animals? There's an interesting video that I want you to go and look that basically traces the early development of a newt, the red-spotted newt here in the United States. And it will show you in that video, and it's sped up, it's a time lapse and it's sped up, but it will show you what happens as the zygote undergoes cell division and creates a morula. And a morula is just a ball of cells, but that ball of cells is hollow. And then as those cells continue to divide more and more and more, those cells get smaller and smaller, and now the ball is hollow. There's space inside the ball, and it's just basically a hollow ball of cells. But then that hollow ball of cells begins to invaginate on itself. And that invagination is called gastrulation. During this process, cells will begin to go through differentiation. They will have a fate that is determined. This cell here will become a nerve cell. This cell over here will become a muscle cell. And in organisms that have what we call determinate cell fate, the fate of the cell is determined relatively early in the development of the organism. In indeterminate fate organisms, the fate of the cells are not determined until much later in development. And so this is something of a continuum, but these would be the two ends of the continuum. So when we think about this process of going from zygote to morula to blastula, this is what it looks like. In the protozoans, not the protozoans, in the protostomes, you have the embryo dividing. That embryo develops into a hollow ball, and then that hollow ball begins to invaginate. And when it invaginates, the inner portion here is going to become endoderm, the outer portion is ectoderm, and then you begin to have mesoderm arising from ectoderm on one side and endoderm on the other side, and that's what begins to form this coelom. In this case, this opening, this initial opening, the blastopore, becomes the mouth of the organism. In deuterostomes, you have this early development. You have blastopore formation, and this blastopore, once again, will form endoderm on the inside and ectoderm on the outside, but the mesoderm begins to develop as an outpocketing from the endoderm. And in this situation, that endoderm will then begin to fill up this space and form these openings, but it's not because the ectoderm is producing a layer of mesoderm. It's because the mesoderm is growing as an outpocketing from the primitive gut here. And in this case, the initial opening ultimately becomes the anus of the adult organism. In protostomes, that's where you have determinate development, where these cells, their cell fate is determined relatively early on in embryonic development. In the deuterostomes, which are what we are, the development of cells is more indeterminate, so that if you lose a cell early on in development, it will just grow into a normal embryo, as opposed to if you lose a cell that early on in development, then what that does is it interrupts the development of the organism. And so this is why we can do stem cell research in determinate organisms, the deuterostomes. The deuterostomes are all of the chordates and the echinoderms, and we'll see that once again when we get to the phylogeny. So the mesoderm is what you get from muscles and blood vessels. We have different forms of circulatory systems, closed circulatory systems, where all of the blood stays inside of vessels as it travels through the body. Open circulatory systems may have the blood in vessels for parts of its travel through the body, but then it's just open and kind of percolating through the body of the organism in other parts of the body. And we'll look at that more closely when we look at, for example, circulatory systems in insects versus vertebrates. So when we look at the open circulatory system, this is the situation in an insect. There is a dorsal vessel that shunts blood to the head of the insect, but then that blood, that hemolymph, as they call it in insects, is free to percolate throughout the body of the organism, and then when it gets back into the abdomen, it gets pushed back into this dorsal aorta here that then has openings, these ostia, these openings that allow the blood to come back in, and then it gets pumped back up to the front of the body. You, on the other hand, have a closed circulatory system. In the best of all possible worlds, your blood stays enclosed within blood vessels as it travels around your body from your heart, taking it out through what we call afferent arteries that carry blood, oxygenated blood, away from the heart to the extremities, and then that oxygen depleted blood comes back to the heart through the efferent veins, which are bringing that deoxygenated blood back to the heart. Then the portion of the heart pumps it out to the lungs, comes back to the left ventricle, and then it's pumped out again. But it stays inside of vessels all the way down to the capillaries in the tips of your fingers or the tips of your toes, or the capillaries that serve any of the organs in your body. Another characteristic or another evolutionary development that happens within the animals is the development of segmentation. And segmentation provides a couple of advantages. It allows redundant organ systems in adults, such as what occurs in annelid worms. And so we'll see that in lab, and we'll also see it in lecture when we get to the annelid worms. And it also allows for more efficient and flexible movement because each segment can move independently. We see this especially well in things like snakes, where the serpentine movement of a snake is largely due to the fact that the body is segmented, and we have muscles that act within each segment, and then we also have muscles that act across segments. And segmentation isn't a synapomorphy. It's actually appeared several times in the evolution of the animals. So traditional classification of animals, we have multicellular animals or metazoans that are divided into 35 to 40 distinct phyla based on shared anatomy and embryology. Metazoans are divided into two main branches: the Parazoa, which lack symmetry in tissues, and the Eumetazoa that have symmetry in tissues. So that's a major distinction there. And then we have, within the Eumetazoa, we have diploblastic organisms that have 2 germ layers, and triploblastic organisms that have 3 germ layers. And this is the grand scheme of things. We're going to start off talking about sponges, and we're going to slowly march through the phyla, talking primarily about the sponges in the phylum Porifera, the cnidarians. We will talk about bryozoans, annelid worms, mollusks, nematodes, tardigrades, arthropods, onychophorans, echinoderms, and chordates. And so we're not going to talk about every phylum that is available because, we'll talk about Platyhelminthes also, but we're going to talk about those things that are in some cases of medical importance and in some cases just important because they represent particular examples of how evolution has solved particular challenges. And I think with that, we're going to stop because we're going to pick up with sponges on Wednesday. Thanks for coming and listening. And once again, my apologies for not being in class today. My plan is to be in class on Wednesday because the issue that came up today will not come up again. And we, of course, have lab all week long where you will be looking at animals. So have a good night, and I will see you on Wednesday.